Good morning, everyone. My name is Isobel Harrison, and I'm a researcher on the SINAPPS 2 trial. Welcome and thank you for joining us on the fourth presentation of our webinar series on the role of inflammation in psychosis. Before I hand over to today's speaker, I would like to give a few details regarding the webinar. The presentation will last for 20 minutes and will be followed by 10 minutes to answer your questions. As all attendees are muted to avoid background noise, please use the Zoom chat facility to submit your questions. Should we not manage to answer all the questions during the webinar, we'll collate them in a document and distribute in a few days' time. Please note that this and all other webinars will be uploaded on our YouTube channel and on our website, so they can be accessed at any time. So without further ado, I'm delighted to introduce today's speaker, Professor Angela Vincent, who is Emeritus Professor of Neuroimmunology at the University of Oxford and Honorary Professor at UCL. She has spent 50 years researching the role of antibodies in neurological diseases. Professor Vincent will give a presentation entitled Maternal Immunity and Neurodevelopment, a Neglected Field. Over to you, Professor Vincent.
I need to get rid of the faces. That's fine. Okay, so I'm going to talk about this field, which I've been interested in for rather a long time. And it will refer quite a lot to last week's speaker, Stukutina, and you'll hear about her work towards the middle and end. But it starts really with what I started with, which is myasthenia gravis, because that taught us a lot about autoantibodies and neurological disease. In the 1970s, when I started, this was what myasthenia could be like. It was still a very severe disease in many cases. It was usually treated with anticholinesterases, but immunotherapies were not yet regularly used in the 1970s. And early on, the cause was unknown. But within 5 years, the cause had become known, and that was very exciting. And it turned out that there were antibodies to the acetylcholine receptors. It could be detected in the serum of the patients, and they went to the neuromuscular junction and they caused loss of the acetylcholine receptors. So in myasthenia gravis, acetylcholine receptors antibodies lead to loss of receptor and effective muscle activation. And you can see in this diagram that the important role of acetylcholine receptors is to receive the acetylcholine, which is released in vesicles, from the nerve terminal, and binds to this receptor protein, and that activates the mechanism which within milliseconds leads to contraction of that muscle fiber. So most of the acetylcholine receptor antibodies turned out to bind to the alpha subunits, and the acetylcholine receptor has 5 subunits, but the alphas are 2, and therefore antibodies to those alpha subunits can actually cross-link the receptors. And that's one of the main mechanisms that lead to their loss in normal myasthenia gravis. So that was very important. We were beginning to learn a lot about the receptor. And within 5 years, we knew this, which was that this was an antibody-mediated neurological disease. And of course, that is now relevant to a whole host of other conditions in the central nervous system and the peripheral nerves. And the criteria are that the antibodies bind to the extracellular domain of a membrane protein. They cause loss of the protein and or damage to the tissue. Patients improve with plasma exchange or drugs that reduce antibody levels. And the transfer of patient IgG to mice can cause similar clinical features, although these are variable and certainly are not very convincing for some of the central nervous system antibodies we now know about. But what about development? So most myasthenia gravis patients have normal babies. Some babies are born with a transient form of myasthenia gravis due to placental transfer of the acetylcholine receptor antibodies. But occasionally, something called arthroglyposis multiplex congenita, or AMC, occurs, and this is lack of fetal movement of any cause, and there are hundreds of genetic causes of this condition. But they are not always genetic. And this is one of the babies, in fact, it was the first baby we studied, who was the fourth child of this mother. You can see the family history, who had 4 consecutive pregnancies with this condition. And that's very unusual for genetics. It would be very surprising indeed. If you look at the baby's fists are clenched, the knees are fixed, the ankles are fixed in that position, and there's a very small chin, and the baby is not breathing properly, and in fact died shortly after birth. So this is something, probably in the mother's circulation, that causes lack of fetal movement during the pregnancy, and that results in this arthroglyposis. And in fact, she had myasthenia gravis diagnosed very late in her case. Wasn't typical myasthenia, but she had high AChR antibodies. She was treated with plasma exchange, thymectomy, and steroids. And subsequently, she had a son who was very, very slightly affected, and as far as I know, has done very well and is normal. So what was special about the antibodies in that mother? Well, it turned out that she and the others that we studied about that time had an excess of antibodies that bound only to the fetal form of the acetylcholine receptor. And the fetal form differs from the adult form, which is shown here, has an epsilon subunit there. The fetal form has a gamma subunit there, and they're slightly different. And the antibodies in those patients whose babies have arthroglyposis bind very specifically to a site which is shared between the gamma and the alpha, not this site, because this is a delta and an alpha. And those antibodies block the acetylcholine binding, and they paralyze the baby. So these antibodies are like the mother having curare and pumping that into her babies, which make them paralyzed. And the paralysis, loss of movement, leads to the arthroglyposis. In humans, in fact, the fetal acetylcholine receptor is present until around 33 weeks gestation. And after that, it's mostly the adult form. So the antibodies would no longer be effective later on during pregnancy. It's the mid-pregnancy term that is most affected by these antibodies. Now, we actually set up an animal model of this. And at the time, and we're talking now about the 1990s, and that was very simple, really. All you do is inject the IgG or the serum or plasma even into pregnant mice and then look at the effects on the babies. And you can see on the left an arthroglyposis baby, all crunched up, fixed joints, not moving, not spontaneously breathing. It would die immediately after birth. Whereas healthy babies, of course, of baby mice, are moving, their joints are fluid, and flexible, and they survive very well. And it was a very striking animal model. Why did we make this animal model of arthroglyposis? Well, we wanted to answer a question. And that question is, could there be maternal antibodies to neuronal proteins in neurodevelopmental mental disorders? And the first attempt we made, or at least actually it was the second, but let's look at this one, was published, in fact, in 2003 as a short report. And it was a rather unique case. This mother had had 3 children. Child 1 was normal. Child 2 had an autism ASD spectrum type disorder. And child 3 had a language disorder, which in fact tended to improve with time. The mother had antibodies to neuronal antigens in tissue, and you can see the pink staining here, which means that there's an antibody in the mother's serum binding to rat, in this case, neuronal tissue, and you can see these are neurons. She also had, the antibodies also bound to the surface of a neuroblastoma cell line, which suggests there was an antibody to a surface protein, which is so important. And when we transferred the disease using the animal model, we were able to see changes in development, which is shown here compared with control, and also in various tests of coordination, which were significant. And we also did probably the first MRI of brains from a mouse model, and were able to show that there were changes in MRS, both in the choline and the creatinine levels in the affected mice. So it was rather exciting, and we thought we'd get some grants to take this work further. Unfortunately, it was quite impossible at the time because everybody was thinking of genetics, not of antibodies. But there were, in fact, subsequently many publications by others on antibodies to neuronal proteins in mothers of ASD children. ASD, of course, is a rather wide diagnosis, and one had to question whether all the children really had a proper autism diagnosis. Moreover, the maternal sera that they used were usually years after birth of the child. They found antibodies to proteins in the fetal brain preparations, but they usually, well, in fact, they always used techniques that would not necessarily detect antibodies to surface proteins, and they did not try to confirm that their antibodies bound to surface proteins. So it was unlikely that those antibodies were actually causing any of the defects in the children. There's a nice review by Katja Marks with Esther Cudinho and myself, which came out last year, and it's a very systematic review. So if anybody wants to look at the data, it's all covered absolutely systematically in that. Katja is a medical student and still is a medical student, and she did an enormous amount of work on this review. But in fact, we knew that there were possible candidates for antibodies in ASD and other neurodevelopmental disorders. We now know that there are antibodies not only, to specific neuronal and glial proteins in patients with different forms of autoimmune encephalitis. And we also know that mutations in Nmda receptors and Caspr2, another protein that we've all worked on, are found in some patients with rare forms and autism and other neurodevelopmental disorders. So it seemed logical to look for those antibodies and other antibodies that are found in autoimmune encephalitis, such as Esther Coutinho showed you last week, to see whether those antibodies were present in the mothers of our ASD children. And we were able to look at a large gestational cohorts of mothers of children with subsequently diagnosed intellectual and motor disabilities, including autism. And we were comparing with age and parity matched control mothers taken during gestation. So first of all, on the top right, you can see the results from Caspr2 and Nmda receptor antibodies in autism. Actually, there were children with autism, I think, rather than ASD, the wider diagnosis. And you can see that we actually didn't find any increased positivity at all of those two antibodies compared with the control cohort. Actually, we started with mothers with subsequent psychosis and individually matched controlled mothers. So mothers who developed after birth psychosis that they had not had previously. And we did find an increase of Nmda receptor antibodies in those patients, but they were a very small number, only 11 of them compared with 176 controls. So that was interesting, but probably not necessarily relevant to their babies. What was much more interesting was when we actually put all the data together, we found that both Caspr2 and Nmda receptor antibodies were relatively frequent in mothers of children with any of these diagnoses compared with the controls, but this was only significant for Caspr2 antibodies. So could we actually produce an antibody model such as we'd used with the previous experiments? And Esther did a fantastic model using the Arthroglyposis animal model basic protocol to look at the effects of Caspr2 antibodies on neurodevelopment, not only at birth, but over the adult lifespan. Excuse me. And it's a complicated set of experiments, but they were very thorough and beautifully systematically done. So first of all, were the offspring affected? Obviously. The offspring were completely normal in early developmental tests. But after weaning, which is 21 days usually, they gained weight and behaved normally in general tests, although they did show a tendency to prefer dark to light, which is a sort of sign of anxiety. However, at 4 to 6 months, there were clear abnormalities, particularly in the reciprocal interaction test, which you can show here, in nesting, which is a sort of natural behavior of mice. And there was some trend towards a reduction, so a decrease in short-term memory in the T-maze alternation test. So there were certainly some effects on the offspring 4 to 6 months after they'd been exposed to the antibody. So were their brains affected in any way? Could we see any effects on the brains? Post-mortem, which was not done until 8 to 10 months after the maternal exposure to the Caspar-2 antibodies, there were different layers of the prelimbic, infralimbic, and somatosensory cortices, were all contained increased activated microglia. So something had certainly happened in those early exposure, during the early exposure, and had persistent long-term effects. I mean, it is now known, it was then actually, that microglial activation can persist for a long time. But you can see the very striking results in almost every layer of the cortices. And that just shows you that those changes in the microglia are activation. Moreover, the same brain areas had reduced numbers of PSD95 synapses, so reduced numbers of synapses. And these were glutamatergic synapses that were reduced. The inhibitory synapses did not seem to be significantly affected. So that was published in 2017 in Acta Neuropathologica. Somewhat similar data had already been published by that time by Brimberg from Betty Dowman's lab in New York in 2016. And actually, collectively, we were effectively confirming each other's data. So that was interesting. Caspar-2 antibodies could play a role in some form of neurodevelopmental disorder. I don't think one should be too specific, and it may not be autism. What about the Nmda receptor antibodies? Well, obviously they were a very attractive group to look at, attractive antibodies to look at, because so much now is known about the disease that occurs in young women, so those who are childbearing age almost entirely, and apart from the children. And so it was very much a possibility that those antibodies could contribute to neurodevelopmental disorders. And that study was done by the Prus group in Berlin and published, sorry, I haven't got the date, I think it's either 2018 or 19, perhaps 2019. The results are a little bit less well, not less convincing, but a little bit less clear, I think, because they did find quite an effect on the body weight. So the mice were overall not so healthy at birth. They survived less well, and that shows you the survival. In fact, at this stage, and with this dose of the injected antibody, and I didn't say, but this was a monoclonal antibody to the Nmda receptor, to the NR1 subunit of the Nmda receptor. At this stage, there were really quite a lot of deaths. And they put this down to the fact that those animals had a higher pH, but I don't really understand what that means. And they didn't, I think, explain very well what that might mean. When they did some of the scoring of the neurodevelopmental scores, which were perfectly normal in the Caspar-2 model that we did, there was really a very striking and bizarre effect of the antibodies. And some of the control groups, or the GFAP group, for instance, actually had something rather similar. So I'm not quite convinced what this all means. And the long-term effects may be all related to the abnormalities during development, during this early stage, or they may be quite independent and due purely to the Nmda receptor antibodies. It's not clear to me. So in summary, there is increasing evidence that antibodies to neuronal proteins can cause abnormalities in mice by maternal transfer during development. Caspr2 and Nmda receptor are the two most likely targets, but there could be many others that have not yet been identified. The crucial tests are presence of antibodies to neuronal or glial surface proteins and demonstration of pathogenicity in mouse or perhaps rat models. However, there's one thing I wanted to add to this, is how do you treat these mothers, supposing you decide that you could treat them? Well, we know that the IgG crosses the placenta mainly from around 16 weeks. In arthroglyposis, the adult AChRs, as I mentioned, replace the fetal AChRs from around 30 weeks. Plasma exchange and IVIG are in repeated cycles, can protect babies during the critical period. We know that from unpublished data. And what you do is you do ultrasound to monitor fetal movement. So with the arthroglyposis problem, that is relatively easy to treat, but very expensive and has its problems. Placental transfer depends on a fetal FcRn that transports the IgG antibodies. So could we prevent transfer of antibodies with an anti-FcRn? And that's the last slide. Well, it's not the last slide, but this is the model that Esther Coutinho and colleagues at King's and at UCB have recently published. And what it shows is that if you use a monoclonal antibody to the FcRn during pregnancy in the animal model, you can reduce the IgG and the AChR antibodies, quite significant in the fetus. You have less deformities in the mice and more spontaneous movements, although that didn't quite reach significance. But there's a clear evidence that you could reduce the transfer of pathogenic antibodies. And there's Leslie and Esther Coutinho. Leslie did all the sort of injection side, and Esther did all the organization and most of the behavioral testing on those animals. So in summary, there is increasing evidence that there are antibodies. Caspr2 and Nmda are the most likely targets. Crucial tests are the presence of antibodies and demonstration in mouse and rat models. And there should be a line which is saying these are potentially treatable forms of disease. Thank you. I'm about 3 minutes late, sorry.
Don't worry at all, Professor Vincent. Thank you very much for an absolutely fascinating presentation. We will proceed to any questions, and we have one from Dr. Saif Huda. Thank you, Angela. I really enjoyed that. Do you think the relative scarcity of AMC relates to the treatment of MG, the frequency of fetal AChR in adult mothers, or a combination of both? Also, are we postulating that neuronal antibodies are potentially present in asymptomatic fashion in mothers of babies who have ASD-like disorders? Thank you, Saif.
Well, thanks, Saif. That's two good questions. So the scarcity of AMC relates to partly to the treatment of myasthenia. I think we have less neonatal transfer of antibodies causing transient myasthenia in the babies now than we used to. AMC, of course, is really very rare, but I think it occurs because for some reason, those mothers develop very specific fetal antibodies that are almost invariably present at much higher levels than their adult AChR antibodies. Now, why do they develop those antibodies? We don't know why, but it is a possibility that some mothers are more prone to develop fetal antibodies to their fetal antigens, because we know that fetal antigens do reach the maternal circulation. That's known for several different antigens and genes. You can see DNA from fetal DNA in mother's circulation, for instance. So it may be that sometimes the mother has a normal birth initially, and it may be that normal birth sort of predisposes or partly immunizes the mother to fetal antigens. I think there are all sorts of complexities with that. Then HLA may come into it because the fetus will have a different HLA dependent partly on the father, of course. And there may be some antigens, not the receptor, that are different between maternal and fetal, maternal and paternal, that might therefore contribute to making the mother feel that what the fetal protein is foreign and she should react against it. So that's one possibility. The other regarding autism and other neurodevelopmental disorders is, yes, I think that we should be aware that there could be antibodies to all sorts of different proteins in these disorders. And there is, in my view, a need to look much more closely for antibodies to all sorts of proteins. I mean, proteins that are involved in neuronal migration, for instance. Anything that affected neuronal migration could easily be causing or predisposing to neurodevelopmental disorders. So I hope that has answered your question.
Thank you very much. We have, Saif says thank you. Sorry to mispronounce your first name, Saif.
Oh, I probably mispronounce it. I've always called him Saif, not Saif.
Okay.
I've known him a long time.
We have a question from somebody in our research group asking if there are any recommendations for clinical practice in terms of this work.
Well, I think, I mean, if you go back to the AMC, which I have to go back to because it has now become quite regular practice for geneticists looking at histories of arthroglyposis in their clinics to think, well, should we be looking for an antibody? Should we test for the acetylcholine receptor antibodies? And in fact, you can pick these patients up, the arthroglyposis ones, just by doing the conventional myasthenia gravis antibody test, because we have always insisted that that test should contain fetal acetylcholine receptor as well as adult acetylcholine receptor. And so when you test for antibodies, you actually pick up the fetal specific AChR antibodies. And that is a way of identifying mothers who might be at risk of having babies with arthroglyposis. Most mothers who have myasthenia have perfectly normal babies or very slightly affected transient ones. But there's also, as it happens, and this could be relevant to other conditions, a growing recognition that sometimes mothers, even without clinical myasthenia, actually have antibodies to the acetylcholine receptor, which can affect the babies, not necessarily by causing arthroglyposis per se, but by causing a myopathy post-birth. And this is something which I am looking at now quite closely with colleagues in Ireland and also in London, and collecting these mothers' samples and the histories of their children, because they have consecutive pregnancies affected with a myopathy. And we believe that this is going to be an antibody-mediated form of a new disease, which they call Faris, but I won't go into. I was going to put something in about it, but it's too short to talk. So there may be long-term effects of antibodies we don't know about. And some of those, the mother may be completely, well, many of them, the mother may be completely normal. That's the problem. You're looking for something in a normal mother. So the question obviously is, well, what is the cost benefit of testing lots of people for these antibodies? And of course, at the moment, there's no question of doing it. But in the arthroglyposis genetics clinics, they will do it if they have any suspicion that the mother's, well, particularly if there's consecutive children involved, for instance.
Thank you very much. Thank you. Okay, we have reached the end of today's webinar. Thank you all for attending. We hope you found the presentation interesting and informative. And we would like to invite you to join or to watch our webinars from this series, which will all be available shortly on our YouTube channel, which also contains the webinars from last summer's series. So this summer series, I'd like to thank Dr. Jonathan Rogers, Dr. Sarah Crisp, Dr. Esther Coutinho, and Professor Angela Vincent for a fantastic series of talks. The link to our YouTube channel is on our website, which is www.synapps.org.uk. We will be sending out links to these talks as well. So thank you, everyone, and enjoy your day.